Good afternoon, everyone. Welcome to the Clark T. Sawin Historical Vignette. My name is Don St. Germain, and Val Galton and I co-chair the ATA's History and Archives Committee. The presentation and discussion today recognize that 2017 is the 50th anniversary of the publication in the Journal of Clinical Endocrinology and Metabolism, a report by Sam Rafatoff, L.T. DeWind, and Les de Groot, describing a familial syndrome of apparent resistance to thyroid hormone. It's difficult to overstate the extraordinary importance of these key observations, as they have served as a catalyst for many critical discoveries and insights into the mechanism of thyroid hormone action and the effects of T3. To celebrate this 50th anniversary, we have asked Dr. Peter Kopp from Northwestern University, the editor of the journal Thyroid, and an outstanding scientist and clinician in his own right, to organize for us a conversation with key leaders in the field of thyroid hormone action and resistance in order to highlight the extraordinary significance of this event. We are indeed indebted to Peter for his insights and dedication in putting this program together. So without further ado, I will turn the program over to Peter and our panelists, whom he will introduce. I extend to our participants the association's great appreciation for their participation today. Peter?
Dr. Saint-Germain, ladies and gentlemen, members and guests, good afternoon. I'm deeply honored by the invitation of the History and Archives Committee and the Program Committee to moderate this year's Clark Sawin Historical Vignette, which always forms one of the highlights of the ATA meeting. It is, among others, the merit of personalities like Clark Sawin and Lynn Loriot that the history of endocrinology and thyroidology continues to be cherished and presented in a meaningful format. So, as Dr. Saint-Germain emphasized, 2017 marks the 50th anniversary of the discovery of the syndrome to thyroid hormone. Thus, it is appropriate to ask the questions, where do we come from? Where are we, and where are we going? We chose to do this in a format of a dialogue, including the key protagonists involved in the elucidation and characterization of RTH, Dr. Samuel Rafatoff. Dr. Gilbert Daniels from the Massachusetts General Hospital will comment on Fuller Albright, who was active at his institution. Douglas Forrest from the National Institute of Health, who has been working in Björn Wenström's laboratory and who has generated numerous mouse models, will comment briefly on the cloning of the TRs. Last but not least, Carla Moran from the University of Cambridge, who has been deeply involved in the characterization of the RTH alpha phenotype, will provide some insights into this novel form of thyroid hormone resistance. It is my hope that some of their personal perspectives will illustrate and enrich our historical understanding and illustrate the remarkable journey completed during these 50 years. In the 1930s, several clinician investigators speculated that end organ resistance to hormone action could be present in a subset of patients with phenotypes resembling, at least in part, hormone deficiencies. The most prominent among them was the legendary Fuller Albright, whose genius insight into numerous aspects of endocrinology have been presented by Dr. Lynn Loriot at the Clark Sawin lecture in Phoenix in 2006. Dr. Daniels, could you briefly comment on Dr. Albright, please?
Is this slide going to go on here or not?
Do we have slides?
That's fine. It's easy to argue that Dr. Albright was the most important endocrine clinical investigator of the 20th century. And that's particularly striking since his career was tragically cut short in his early 40s by the ravages of Parkinson's disease and its treatment. Dr. Albright was the first North American to describe hyper and hypoparathyroidism, and his investigations into calcium metabolism were legendary. It's less well known that he was the first person to actually formulate the correct pathophysiology of Cushing's syndrome and the adrenogenital syndrome. The younger people in this audience may not realize that Klinefelter's was described by Dr. Albright and the name attributed to the fellow who got to present the case at one of the national meetings. Dr. Albright's group provided the most meticulous description of Turner syndrome, and he was the first to show that the FSH was dramatically elevated in that syndrome. He was the first to describe renal tubular acidosis, bone loss from immobilization, the milk alkali syndrome, and vitamin D resistant rickets. He was the first to use estrogen to treat postmenopausal osteoporosis, to treat growth hormone excess, and to suppress ovarian function, which ultimately led to oral contraceptives. His name lives on in two syndromes which are well known, Albright's hereditary osteodystrophy and the McCune-Albright syndrome. But the Forbes-Albright syndrome, which is hyperprolactinemic amenorrhea, is no longer use that term. In a CPC describing a patient with renal cell carcinoma, Dr. Albright was the first to hypothesize that tumors could could ectopically produce hormones, in that case, the ectopic production of PTH.
Thank you. And what was his specific contribution to the discovery of hormone resistance?
In 1942, Dr. Albright reported 3 cases of what he called pseudo-hypoparathyroidism. These were called pseudo-hypoparathyroidism because they resembled hypoparathyroidism, but were resistant to the parathyroid hormone. His classic manuscript was Pseudo-Hypoparathyroidism, an example of Sebright-Bantam syndrome. He wrote, theoretically, one could obtain essentially the same clinical picture from failure of an end organ to respond to a hormone as from decreased production or absence of the same hormone.
So can you enlighten us about this funny bird, the Sebright bantam chicken, and how it led Dr. Albright to develop the concept of hormone resistance?
So Dr. Albright wasn't always right. He misspelled Sebright bantam. The Sebright bantam chicken is a male rooster producing testosterone, but instead of having the typical rooster feathers, it had actually female type feathers. And at that time in Dr. Albright's laboratory, they called any clinical syndrome that they thought represented resistance to a hormone an example of the Sebright-Bantam syndrome. And so he called pseudo-hypoparathyroidism an example of resistance to testosterone action.
So let's leave the chicken and get back to the patients with resistance to PTH. This slide illustrates one of the patients from Dr. Albright's publication. This 28-year-old woman was referred to the MGH with the diagnosis of hypoparathyroidism and epilepsy. Her physical exam is remarkable for short stature, a round face, and short metacarpalia 4 and 5, a phenotype that we now describe as Albright hereditary osteodystrophy. Among other findings, she also had signs of hypocalcemia, such as a positive cross-check and Trousseau sign. Dr. Daniels, what were the biochemical findings leading to the diagnosis of pseudohypoparathyroidism or resistance to parathyroid hormone?
The 3 cases he described had symptomatic hypocalcemia and hyperphosphatemia. He administered parathyroid hormone to all 3 patients and found that the calcium did not rise and the phosphate did not fall. And note, this was a time before parathyroid hormone could be measured. He also ascertained that these patients had never been exposed to parathyroid hormone, so they didn't develop a secondary resistance, and he therefore concluded they must be resistant to the action of parathyroid hormone.
So very briefly, was Dr. Albright's hypothesis that the Sebright phenotype is caused by diminished testosterone action correct?
No, he was incorrect because the defect in the Sebright chicken is that they produce testosterone, but they have an abnormality of aromatase and have increased production of estrogen in the skin. If I may, I'd just like to end with my favorite Albright thyroid anecdote, since this is the ATA meeting. He was participating in a symposium on multinodular thyroid. The surgeon described his first 10,000 cases of thyroid surgery. Another one described his first 5,000 cases of surgery for nodular goiter. Dr. Albright stood up and said, "I saw a case of nodular goiter, and I'd like to share my insights."
So with this background, let us move forward from 1942 to the 60s. This slide shows Dr. Samuel Refetov as a resident in internal medicine at the Good Samaritan County Hospital in Los Angeles, where he was active in 1964 and early 1965. Handsome, I hear a lot of reactions. So, Dr. Refetov was born in Ruse on the Danube River in Bulgaria. He then emigrated to Belgium as a teenager, where he obtained his high school diploma from a French-speaking high school in Antwerp. A second migration led him to Montreal, Canada, where he received a baccalaureate degree 1959 from the University of Montreal. He then completed medical school at McGill University in 1963, and subsequently he was an intern at the Hôpital Notre-Dame in Montreal and a resident at the Hospital of the Good Samaritan in California from 1964 to 1965. At that time, in 1962, a 6-year-old girl was brought to the emergency room of the Los Angeles County Hospital for the evaluation of injuries after she had been hit by a truck. Dr. Refetov, what happened next?
The attending physician, Dr. Lauren DeWind, saw that the X-ray was unusual and was puzzled by the findings. He also ordered plasma-bound iodine, PBI, as a measure of thyroid hormone. He then encouraged me to further characterize the unusual phenotype.
So can you briefly describe the phenotype and the X-ray findings of this index patient?
So she had deaf mutism, and her thyroid gland was slightly enlarged on palpation. She had short stature, bird-like facies, pigeon breast, and winged scapulae. The radiological survey disclosed steeple epiphysis of the major secondary ossification centers. Since some of these findings are typical of neonatal hypothyroidism, measurement of her protein-bound iodine, PBI, in serum was obtained, which at the time was the routine test for the estimation of the serum concentration of thyroid hormone. Surprisingly, the value was high. Though compatible with the absence of other signs and symptoms of thyroid hormone deprivation, the high PBI value was not in agreement with the lack of manifestation commonly associated with thyroid hormone excess. We thoroughly characterized the phenotype of the patient before I moved to Boston in July 25th, 1965. I didn't know that the date will be put in, the year was enough. To the Lay Clinic, and then after that to the Brigham. However, the work on the index patients and the Gonzalez family continued.
So in this letter from August 3rd, 1965 to Dr. DeWind, Dr. Refetov commented, and all these letters are in the archives of the ATA, so you can look at them there. Well, right now they're in my office at home. That time permitted me to think and work up different hypotheses in an attempt to explain the possible abnormality of the thyroid metabolism in the family we are dealing with. And you also mentioned Dr. Les de Groot, who initially was supposed to be part of this session, but who unfortunately could not attend, who was in the thyroid unit of the Mass General Hospital. So what was his role in the studies of the Gonzalez family?
So Dr. de Groot was key for the completion of the iodide isotope kinetic studies. In early August 1965, he sent me back to Los Angeles on my own expense with a mixture of 125 and 131 iodide that I held in my pocket when I mounted the plane. Incidentally, there was no, at the time, there was no TSA. After injecting the labeled isotope to the eldest affected individuals, and that was in August 7th, 1965, the Watts riots broke up in August 11th. As the family lived in the Watts area, Dr. Peter Sargent, the medical director of the Good Samaritan Hospital, arranged me to be escorted by 4 National Guards to collect blood and urine each day until August 16th, when the study finished.
So at this point, let's summarize the findings obtained from the Gonzalez family. Dr. Rafetoff.
So the index patient, here shown with the red arrow, she was the offspring of first-degree cousins once removed. Further analysis identified the same phenotype in 2 brothers. Hence, the mode of inheritance was expected to be autosomal recessive. The results in the children were rather unexpected. The children had not only normal metabolism, but also had none of the known inborn error of thyroid hormone metabolism symptoms. In aggregate, the data suggested that the disorder was due to a partial failure of the body tissues to recognize thyroid hormone, further termed the syndrome of generalized resistance to thyroid hormone.
So an abstract, which was very well received, and even the press got interested, was present at the Endocrine Society meeting 1966 in Chicago. And the first paper describing the novel entity on a possible target organ resistance to thyroid hormone was then published 50 years ago in 1967. So what was your speculation about the mechanism underlying the resistance?
So in the discussion, we stated that the most reasonable interpretation is an inability of the hormone to pass from blood to cells or target organ refractoriness to thyroid hormone. Of note, at the time, we did not know anything about uptake of thyroid hormone by specific channels, nor did we have any understanding how thyroid hormone works exactly at the cellular level.
So what happened in the subsequent years?
The clinical features of numerous other individuals and families were carefully delineated. The syndrome was defined as reduced responsiveness of target tissues to a supply of thyroid hormone that under normal circumstances would be excessive. The cardinal features include free thyroid hormone levels that are unable to normally downregulate the secretion of TSH or to produce clear symptoms and signs of thyroid hormone excess. In most instances, affected individuals were found to have a goiter. Contrary to the recessive transmission in the key family, transmission of all other families appeared to be autosomal dominant. A high prevalence of hyperactivity, learning disability, as well as mental retardation of variable severity were also observed. The puzzling part was the lack of uniformity in the clinical manifestations. The phenotype spectrum led to the clinical subclassification of the syndrome into generalized and selective pituitary resistance to thyroid hormone, and to the conviction that RTH encompassed a heterogeneous group of conditions with defects residing at different steps involved in thyroid hormone mediation of thyroid hormone action.
So in the 1960s, Jamshed Tada in the UK and colleagues showed in several publications that T3 stimulates RNA synthesis and subsequent protein synthesis, indicating that it has a transcriptional effect. Here is one of these experiments demonstrating a rapid increase in RNA levels in the liver of thyroidectomized rats after the injection of a single dose of T3. Dr. Rafetoff, was the next crucial step in our understanding of thyroid hormone action?
In 1972, Jack Oppenheimer and his colleagues, incidentally, Jack Oppenheimer's application for grant at the time had 120 pages, and colleagues demonstrated the presence of putative nuclear thyroid hormone receptor. This figure shows the binding of radioactive T3 to the nuclear fraction of rat liver cells and the decrease of the radioactivity with concomitant injection of increasing amount of non-radioactive called T3.
So experiments with white cell nuclei from RKH subjects then showed significant reduction of the affinity for T3 in some subjects, but not in others. Moreover, there were discrepant results between affected subjects belonging to the same family, and in aggregate, the results remained inconclusive. So the next breakthrough had to await the cloning of the thyroid hormone receptors 13 years later in 1986 by Björn Wenström's and Ron Evans' groups. Dr. Forrest, you have been in Dr. Wenström's laboratory. Can you please comment on the cloning of these receptors?
Yes, so the identification of the T3 receptor came from unexpected quarters, and that was the study of chicken retroviruses and chicken leukemia. So you can imagine, of course, if you send your grant to the study section and say, we're going to try and find the T3 receptor looking at chicken leukemias, you can imagine the response you might get. However, I think this illustrates a very important point, a crucial point that came from this era of research, which is that it's very important to keep an open mind and to keep up with advances in fields that are beyond our own specialization. So specifically, what was going on here, this is the EMBL, European Molecular Biology Laboratory in Heidelberg, 1986. And there were three groups. Björn Wenström's group was part of this cell differentiation program. So the EMBL was a leading center for studying oncogenic retroviruses, which at that period was an explosive field of research in the 1980s.
So one person is missing here. Jan Saab, can you comment on Jan?
So in the photograph that was shown, Björn Wenström, Klaus Damm, Alberto Munoz were shown. Also, Hartmut Boig was a collaborator, wonderful cell biologist, could study erythroid transformation in chick systems, and it was a very close collaboration. Jan Saab, the leading author on the cloning of the T3 receptor, was not in the picture. Here is another picture of Jan. So Jan was a graduate student, so for all the students and postdocs in the audience, take this as an inspiration. Doesn't matter what your position is, if you're in research, you can find something really remarkable. And his task was, Wenström's group had cloned both of the oncogenes carried by this chicken retrovirus, AEVES4, the VRB and VRB oncogenes. The ERB oncogene was a mutant form of EGF receptor, and Jan's project was to clone and try and characterize, identify what did the normal proto-oncogene do for the viral ERB-A oncogene. So the cDNA, they'd cloned it, and Jan was doing expression studies and testing what on earth this protein might do. So another advance that was taking place at this time was that the very first steroid hormone receptors had been cloned that same year. The first, the GR from Ron Evans' group at the Salk Institute, and secondly, the ER, estrogen receptor, by Pierre Chambon's group in Strasbourg. So immediately it was obvious that the viral ERB oncogene had a striking homology with these steroid receptors in the C-terminus and especially in the central DNA binding domain. So this stimulated the idea that the ERB-A product was some kind of steroid receptor. So Jan tested various hormones, none of them bound. And then was a crucial piece of lateral thinking. I think you have to think beyond the confines of the obvious. And he began to look at other hormones. He looked at T3, bingo, found high specificity, high affinity binding.
So we have now here these thyroid hormone receptors. So can you very briefly comment on these two genes and the structure of the protein product most in the audience know this, but what are sort of the canonical features?
Yeah, so the gene that Jan had cloned was basically the TR alpha gene. And at the same time, Ron Evans' group had cloned another similar but distinct cDNA, which ended up being the TR beta gene, THRB. So right away, this was also another conceptual advance. It said there was not just one T3 receptor, there were actually two genes encoding T3 receptors. And extensive studies then over many years showed that these genes encoded different isoforms which were differentially expressed in different tissues. I'll just mention the alpha gene encodes one T3 receptor, TR alpha 1. The beta gene expresses two major isoforms, the B1 and B2 isoforms.
Thank you very much. So, Dr. Receptors, once the CRA beta gene was cloned, what was the next step in elucidating the molecular basis of resistance to thyroid hormone?
So using the TR beta gene as a probe, Usala and his colleagues in Bruce Weintraub's group at the NIH could distinguish the paternal allele here shown using restriction length polymorphism that was associated or linked to the RTH phenotype. For example, in the family shown here, the affected individuals carry all the allele, which is marked with a C. These findings suggested that the causal defect is either within the thyroid hormone receptor beta gene or at least in its close vicinity. Steve Usala obtained his BA, MD, and PhD degree at the University of Chicago, and I knew him while in Chicago, and he often communicated with me while doing this linkage analysis. So I learned about the results before they were submitted for publications. However, we are doing the same study in another family and got contrary results, which I shared with Stephen and warned him about the publication. The mistake was actually ours, caused by mislabeling two samples.
So the plot is thickening, and you had moved to the University of Chicago with Dr. DeGroot, here shown on a group picture from 1980. Here is Dr. Refetov, here Dr. DeGroot. And aside from being productive in many other fields, it was now obvious to look for mutations in the TR beta gene in affected individuals from RTH families. Dr. Refetov. Huh?
Indeed, shortly after the demonstration of linkage in the RTH phenotype to the TR beta locus, our group and that of Weintraub could demonstrate distinct point mutations, those are missense mutations, in affected subjects in two unrelated RTH families.
So what were the hallmarks of these first mutation?
In a T3 binding assay, RT beta wild type exhibited high binding of I-125 iodide labeled T3. In contrast, the mutant lost the ability to bind T3.
So we all know that RTH beta is usually dominantly inherited. So what led to the recognition of the dominant negative effect of monoallelic TR beta mutations, and how could you distinguish between haploinsufficiency and a dominant negative effect?
So the same family, the one identified to have a mutation, G324R, was further characterized in vitro. In a transient expression study, only the wild-type TR activated the RADG8 promoter in a fuse to the cat reporter. Additionally, when the wild-type TR and the mutant TR were co-transfected, the mutant inhibited the acetylation induced by the wild-type TR. Of note, there was no difference in DNA binding between the wild-type and the mutant. From these results, we concluded that the mutant inhibits the function of the wild-type allele in a dominant manner, thereby explaining the clinical manifestation of thyroid hormone resistance produced in a mutant present in the heterozygous state where one normal allele was present. This paradigm was confirmed by Krishna Chatterjee, then working in Larry Jamison's laboratory, as well as by other groups investigating this syndrome.
So over the years then, numerous mutations associated with RTH beta phenotype have been described. And what is the overall lesson from these observations?
Indeed, as illustrated here, close to 200 mutations have been described, with the exception of the index family in which the affected individuals were homozygous for the complete deletion of the THRB gene. The majority have single nucleotide substitution, resulting in single amino acid replacement and less commonly premature stop codons. Within the TR beta, there are located in the T3, but most of the TR beta mutations are located in the T3 binding domain, adjacent, and also in the adjacent hinge region. No mutations have been identified in the DNA binding domain, as such mutations will not have a phenotype because they cannot exert a dominant negative effect. Functionally, they result in variable reduction in the affinity for T3, while DNA binding and dimerization with a homologous or heterologous partner is preserved. They exert a dominant negative effect through an increased affinity for co-repressor or decreased affinity to co-activator cofactors.
So João Anselmo studied 167 members of a Azorian family with RTH, and the affected individuals, shown here in red, had the RTH phenotype caused by the so-called R243Q mutation. This very large pedigree allowed to study the impact of elevated maternal thyroid hormone levels on affected and unaffected offspring. So what were the main findings of this study, Dr. Rafetoff?
The two key findings. There is a higher rate of miscarriages in mothers affected by RTH. This very likely affects primarily those fetuses that are unaffected because of their exposure to high thyroid hormone. And this is because these women gave birth to more affected and unaffected infants. And this contrasts with infants born to normal mothers because their father was affected. In that case, there's equal birth of normal and affected infants, as it's expected in a dominant inheritance.
And so it also has an impact on birth weight. I know the previous session this afternoon was on the impact of thyroid hormone dysfunction in pregnancy, and we can learn a lot of lessons here. So Dr. Rafetoff, what can you tell us here about the birth weight in these infants?
So in addition, the second observation with this family was the non-affected individuals have lower birth weight and suppressed TSH and when born to affected mothers. These data indicate a direct toxic effect of thyroid hormone excess to the fetus.
So what happens to the thyroid axis in unaffected individuals with RTH exposed to high maternal levels of thyroid hormone during fetal life?
So those same unaffected infants that appear to be toxic at birth, when they reached maturity, in this work by Panuda Srikom-Kun, individuals exposed to the high level of maternal thyroid hormone in utero responded less to the suppression of thyroid hormone by the reduction of TSH. And this contrasts with those normal individuals born to fathers who had resistance to thyroid hormone. Mechanistically, this is probably mediated by increased expression of deiodinase type 3 in the anterior pituitary, which enhances the degradation of T3 as demonstrated in mice.
So back to mice. many investigators, including, among others, Björn Wenström, Douglas Forrest, Fred Wandisford, Xiaoyan Cheng, Jacques Samaru, Juan Bernal, Roy Weiss, Frédéric Flamand, and Greg Brent have generated numerous murine knockout and knock-in models. Dr. Forrest, can you tell us briefly how mouse knockouts have enhanced our understanding of the biological functions of the TRs?
Yes, and it's an ongoing story. We don't know the half of it so far, but a few summary points. Life actually goes on in the absence of any nuclear TRs. You can delete them all from mice, and life goes on. Doesn't go on very well, however. There are multiple systems, tissue systems, are defective. In fact, another, I think, conclusion we can draw from many years of study is that probably every single cell type in the body has some type of response to T3. And as I say, we still don't know the half of it, so there is great scope for research. The systems that we really have gained deep understanding in, they include classical systems that are known to be T3 responsive, such as the heart, the liver, bone, for example, and the brain. We have made major insights into many of these systems in understanding how the different cell types respond. We also have unexpected surprises, and a number of systems had previously not really been recognized as T3 response tissues. I'll just mention one is the color visual system, the cone photoreceptors in the retina.
So in aggregate, it became clear that TR-alpha is the predominant subtype in bone, the gastrointestinal tract, cardiac and skeletal muscle, and the central nervous system. TR-beta 1 is most abundant in the liver and the kidney. TR-beta 2 is more discretely expressed in the hypothalamus, pituitary, cochlea, and the retina. Importantly, in the absence of hormone thyroid receptors that are not bound to ligands, repress or silence target gene transcription by recruiting multi-protein complexes containing co-repressors, whereas T3 occupancy results in dissociation of the co-repressor complex and recruitment of co-activator proteins, which mediate hormone-dependent transcriptional regulation. Based on these observations, many speculated about the phenotypic consequences of putative mutations in TR-alpha. However, this was not really apparent. The molecular cause of RTH-beta phenotype has been identified by classic linkage studies and the subsequent detection of mutations in TR-beta. So, Dr. Morin, how has the RTH phenotype alpha been discovered?
Thank you, Peter. So the discovery of RTH alpha was made in a very different way. This is the first patient who was described. She was a 6-year-old girl, and she was referred to my colleague Krish Chatterjee in Cambridge with developmental delay, reported obesity, and mildly abnormal thyroid function. And she also had a skeletal dysplasia, which was manifested by short stature, an increased head circumference, delayed closure of the fontanelles, and epiphyseal dysgenesis. And Krish at the time couldn't identify a candidate gene that would explain all of these features. And so he decided to proceed to whole exome sequencing. And then subsequent analysis of the sequencing results identified a heterozygous mutation, E403X, in the THRA gene, which of course encodes the alpha form of the thyroid receptor. And I know at the time Krish was very excited and immediately suspicious that this was the cause for a number of reasons, primarily because the mutation was predicted to result in premature truncation of the receptor. The mutation wasn't present in either parents, both of whom were phenotypically normal. And of course, he knew of the critical role of TR-alpha in thyroid hormone signaling.
So how did the murine phenotypes help you in delineating the phenotype of this first patient?
Yes, at the time, these were really crucial. And Krish reviewed the patient's phenotypic information to determine whether her features matched those found in the murine models. And there are 4 mutant models which have been produced, which are listed here. And what he found was that almost all of the prominent mouse phenotypes were present in this patient. So as already mentioned, she has growth retardation and delayed bone age with developmental delay, but she also had a mild bradycardia. She had an ataxia, delayed dentition, severe constipation, reduced basal metabolic rate, and an anemia. And I know that at the time he really found the insights reported by Bjorn Wenstrom in his paper in 2008, which just summarized the mutant mouse models, but also predicted what a human patient with RTH alpha would be like, and these predictions proved remarkably accurate.
So in what way do the murine and human phenotype differ?
So there are some differences. Most of the mouse models had reduced body weight and reduced adipose tissue. We find that the patients have obesity. Delayed puberty and breathing abnormalities were reported in the mice, but we haven't found that as a phenotype in patients. And the thyroid biochemistry is different. The mice have a high TSH with normal thyroid hormone levels, and humans tend to have a normal or mildly high TSH, a low or low normal T4, and a high or high normal T3.
So since the identification of this first patient in 2012, a number of other cases have been described. Can you tell us briefly about these mutations found in these cases and what the implications are?
Yeah, so to date, there are over 30 cases that have been described, and 15 different mutations have been described in these cases. The first of these, who was reported very soon after Chris reported his patient, was identified by Theo Visser's group from the Netherlands, and they described a father and daughter with a frame shift and premature stop mutation. And since then, 3 other mutations, which result in premature truncation of the alpha form of the thyroid receptor, have been identified, and the other patients who have subsequently been identified harbor missense mutations. All of these mutations are heterozygous, and they are all within the ligand binding domain of TRA. Some of the mutations affect both alpha-1 and alpha-2 proteins, but with the exception of one case, we haven't been able to identify any additional or altered phenotype in these patients. And I think it's worth stating that having identified this number of patients now, we think that there is a phenotypic spectrum, with patients harboring truncating mutations having a more severe phenotype compared to those with missense mutations.
So thank you very much. There are numerous other aspects, for example, the identification of co-repressor and co-activators, or the crystal structure of thyroid hormone receptor, that could be discussed here. However, time constraints unfortunately force us to move on. I would just like to emphasize that in addition to the discussed receptor defects, we now know several forms of decreased sensitivity to thyroid hormone that are different, including mutations in membrane transporters, MCT8, and thyroid hormone metabolism. And these defects have in part also been characterized by Dr. Refetov and his group. It is also clear that other forms of impaired sensitivity to thyroid hormone await to be discovered. A true catalyst for the worldwide interactions focusing on resistance to thyroid hormone and thyroid hormone action are the biannual workshops on resistance to thyroid hormone. I also wish to acknowledge the American Thyroid Association for supporting several of these events. It's really an absolutely outstanding meeting. The first workshop has been held in 1993 in the Magdalene College in Cambridge. It has been organized by Krishna Chatterjee and Paula Peck-Pakas, who are shown on this slide. Here's Paula, here is Krishna, together with Sam Refetov, Roy Weiss, and João Anselmo from the Azores. This is a picture from the participants of last year's meeting in Colorado Springs, which has preceded the meeting in Denver. And next year, we will have a meeting in the Netherlands that will be close to the ETA meeting. I think it's also important to emphasize that numerous groups and investigators have contributed to the advancement of the field, and I do apologize that they could not be named in this very short overview. Moreover, any list would of course be incomplete. This graph serves as an illustration of the multitude of contributors, and it also symbolizes that exchange, communication, integration, and building bridges rather than isolation and building walls lead to novel horizons. So we have rushed through more than 50 years, highlighting some of the key steps, yet of course omitting quite a few others. It all started with an astute clinical observation of Dr. De Wind and Dr. Refetov, whom we honor for his outstanding contributions to our understanding of thyroid hormone pathophysiology. Now, Dr. Refetov always emphasizes that his greatest professional pride and honor is his role as a mentor of his fellows. Dr. Refetov.
Yes, this important slide lists my fellows and graduate students over 46 years. A total of 56 are listed in chronological order from left to right. They came from 18 countries, 5 continents, 18 became professors, 5 heads of institutes or departments, and 23 students and 8 visiting professors are not listed. But here in the audience, I want to mention my first fellow, Neil Sherberg, is here, as well as David Sarney, that followed, Yoshiharu Murata, Kenneth Ain, Joachim Pollens, Chutin Sripadanang, and Haruki Fujiwara. Thank you.
Thank you, Dr. Refetov. So some question the value of studying rare disorders. But as illustrated by this quote from Archibald Garrett from 1924, the hereditary and inborn departure from the normal of metabolism may throw a ray of light into some dark place of biochemistry. So were Albright here with us today, he would be delighted to see the ramification of Dr. Refetov's discovery and how molecular reductionism can provide fundamental mechanistic insights, which of course can also have impacts on clinical medicine. So ladies and gentlemen, I wish to thank you for your attention and all the panelists for their contribution. And please join me in recognizing Dr. Refetov and his colleagues for their insightful clinical observation and the ensuing outstanding creativity during the last 50 years.